Hi, this is Guy Haskin-Fernald, and I'm going to talk about a pair of papers that combine chemoinformatics with side effects to make some interesting predictions. The first paper is really the paper that describes how they generated an interesting data source of side effects. And this was essentially done by going to the FDA's database of labels and parsing those. They then normalized those using CoSTAR terms, which has now been relabeled as MEDRA, which is essentially just a standardized nomenclature for adverse effects, so that if someone has a headache, you know it's classified the same across all of the different labels that are combined. It also then combined information from PubChem to get structural and chemical information about the drug, so you can make comparisons between the chemicals. They combine these into a database. And one of the first things you see when you combine all this data is that there are these exponential effects. So these two plots are showing the number of side effects per drug over on this side, and the number of drugs per side effect over on this side. And one thing you'll notice is that here's 10, and almost all drugs have at least 10 different side effects listed on the label. Some of them have a very large number, and very few have only fewer than 10 side effects listed for them. This other graph shows that some side effects are on nearly all drugs, and very few of them are on just a few of the drugs. And you can also note by looking at the different codes associated with those, not shown in these plots, but the side effects do occur in different organs, so they're occurring in different parts of the body. They may be limited to the liver, they may be limited to the muscles. Those are categorized here in these ATC categories, where you see alimentary tract and metabolism, for example, blood and blood-forming organs, etc. And what you see in this plot is the categories are what is being treated by the drug, and the indications or the side effect class are listed along this side. And the color is showing you the association. In some cases you see a negative association, for example, endocrine disorders with blood and blood-forming organs for drugs. You see a negative association. In other cases you see positive associations, such as ophthalmic sensory disorders and an association with sensory organs, which makes sense because if you're treating that, you'd expect a positive association. The other interesting thing to note is how they relate to placebos by clinical trials. And in some cases, you see here with this blue line, you see many side effects which occur with equal frequency with the placebo as in a clinical trial. The red line over here shows that with the drug, it is twice as likely. And if you look at them, you can see the ones that are equal frequency over here are very common, such as headache, and the ones that are less, that are more likely with a real drug, which would really be a real side effect, are things like erythema, amenorrhea, leukopenia, things that are more serious and are likely to be caused by the drug. But the really interesting use of this is using it in a biological context. And that comes up in the second paper, which is drug target identification. So a key and interesting problem in the study of pharmacology is identifying drug targets. And using chemoinformatics and pharmacogenetics, you can do things to identify a drug target. And that can be done with things like similarity of chemicals or using docking algorithms. But what this paper asked is, can we use that resource of side effects in order to find a shared target? So what the authors did in this paper is took 746 drugs, derived their side effects profiles from that database, and then found drug-drug pairs which were suggestive of sharing a target. And 261 of those were novel, and then they went ahead and actually validated those by doing experiments. So the first thing to consider in this is, what is an off-target? As far as a drug is concerned, it's just binding to whatever it binds to in the body. In the context of this paper, we consider off-targets to be things that are sort of downstream and sort of unintended. We're not trying to treat that part of the body, but that part of the body is being affected. It could be caused by many things, such as different dosages, maybe because of a pharmacogenetic reason leading to insufficient metabolization, maybe related to active metabolites that are in the body, aggregation. One example of this kind of off-target effect is with minoxidil, which was a drug originally developed as a vasodilator and for its properties for binding potassium channels. But it turned out that due to reasons which are still unknown, it is a stimulant for hair growth, and so the drug has been repositioned and is commonly used for that now. So the way they went about doing this was by, first of all, extracting the information from the labels, and that was what was discussed in the first paper, and then by relating them all using the ontology, and that was the MEDRA system that we talked about as well. They perform a downweighting so that correlated side effects get less weight, because some side effects occur together almost all the time, and they do a similar thing for frequent side effects as well. So the less frequent a side effect occurs, and if it doesn't occur with other ones, then it gets a higher score. The next step in their method is to calculate raw scores of these weights along with the drugs, and figure out which of these scores are significant. And they do this using a permutation. So they randomize the data, and they are able to calculate a histogram and determined a p-value that is empirically drawn based upon the raw score. You can then combine that with chemical similarity using the Tanimoto metric of comparing two chemical fingerprints, and you can make an interesting plot showing along this side the increase in chemical similarity, and along this side an increase in side effect similarity. The color shows going from the red to the green the probability of a shared target. And you can see here with drugs that have very high chemical similarity and very high side effect similarity are in this green zone up here, and they are very likely to share a similar target. If you look at all the pairs that you find, they found 2,903 pairs, and 1,018 of them had a p-value less than 0.1. And they then eliminated pairs that already were known, the ones that were very similar, and ones that did not have human targets, and drugs that were from the same therapeutic category. And they were left with 261 novel predictions that had a p-value less than 0.1. They took out 13 of the most intriguing ones and performed drug activity assays on them, and were able to confirm for these 13 that these tested drugs actually were binding targets that were predicted using their method, simply because the two drugs had similar side effect profiles and a high enough chemical similarity that it seemed likely that they would share a target. And that is it. An interesting paper sort of describing how you can combine side effect similarity with informatics in order to make some interesting biological predictions. Thanks.